Hi, everyone. My name is Sophie Rangarajan. I'm a clinical instructor in neuromuscular neurology, and it's a great pleasure today to introduce Dr. Patrick Shu. Dr. Shu is an assistant professor of bioengineering at UC Berkeley and is also the co-founder and a core investigator at the ARC Institute in the Bay Area. He pursued graduate studies at Harvard University and the Broad Institute, where he worked on pioneering early CRISPR-Cas9 technologies for editing the human genome. His lab currently leverages biological programming, human synthetic biology, and AI to improve human health. And Dr. Shu has received numerous awards and recognitions, including, but certainly not limited to, the New York Times Good Tech Award, MIT Technology Reviews, Innovators Under 35, Forbes 30 Under 30, the NIH Early Independence Award, and the Rainwater Prize for Innovative Early Career Scientists. His talk today is entitled Bridging Biology for AI and Genome Design. So please join me in welcoming Dr. Patrick Shu.
Okay, can you hear me? Wonderful. Well, thank you so much for the kind introduction, and thank you, Stan, for having me here today. And really appreciate all of you coming in person, and also hi to the folks on Zoom. So I'm going to tell you today about some of the work that we've been doing in the lab at the interface of biological engineering, genome editing, and machine learning to try to kind of bring together the fields of kind of AI and biology to think about how we can do molecular design. We'll sort of start the talk by giving you, this is actually my favorite picture. It's the Xerox Alto. It was first released in 1973, so just over a half century ago now. And it was the first desktop computer that was sold with a mouse and a graphical user interface. And it enabled humans for the first time to interact with computers in a simple and intuitive way. And it was actually invented just down the street from where the ARC Institute is today in Palo Alto. And so we've been sort of wondering in the lab, kind of how can we do the same thing with the genome and interact with the foreign language of the genetic code in a kind of a native human interpretable way as well. And so this simple theoretical machine, the Turing machine, was first proposed by Alan Turing, one of the kind of founders, if you will, of modern computer science in 1936. And it's kind of old school, as you can see, but it's surprisingly powerful. It consists of this tape that can store binary information, a head that can read and write symbols, and a set of rules that guides what happens next. If a machine reads a 1, it might write a 0, move left, or change states. And despite this minimal setup, it's been shown that any computer algorithm with the ability to read binary code, think about the information, and move the head to write back onto tape, you can write any computer algorithm, anything from Google search to deep learning using this so-called Turing loop. And so we've been thinking about how we can build an analogous Turing loop for biological information. Obviously we know that one of the most established biotechnologies today involves reading biology across multiple modalities like the genome and epigenome and the transcriptome with multi-omics technologies. And this is how we digitize biological state. And I'll tell you today about some of our efforts to build technologies for thinking and writing about biological information. So, guide RNAs have revolutionized biology by enabling programmability, and some of these foundational technologies like RNA interference or CRISPR technology are fundamentally RNA-guided systems that have this one-to-one search string that allows us to search for short, specific sequences in a really complex genetic code. But the limitation is that none of these really allow for large scale genome writing. And what in principle you would really want with this kind of 3.2 billion letter book of life is a technology that can, instead of changing individual bases or words at a time, you might be able to change entire kind of books or chapters, potentially eventually at the kind of chromosome to megabase scale rather than a few letters at a time. And so there are a few technologies that seem to already do this in nature and do large scale programming, such as transposons, which are the kind of most ancient mechanisms for genetic diversification. And they've been recognized by Barbara McClintock, the original discoverer, as these so-called jumping genes, not as junk DNA as they might have been taught to you in high school, but actually as major players in evolution and speciation. And so I particularly like this excerpt from her 1983 Nobel lecture, kind of undoubtedly new species can arise quite suddenly as the aftermath of accidental hybridizations between two different species, which I think was a really kind of prescient preview to the purposeful hybridizations that we do in modern genome editing. Sort of ends by saying it's difficult to resist concluding that these genetic shocks might have been responsible for the origin of new species. And so yeah, you can sort of see on the right an electron micrograph of a mobile genetic element shown here in this sort of darker kind of color. And it gives you a physical sense of how transposons reassort the components of a genome. And this is actually very personal, right? About half of your own genome is actually derived from these types of transposable elements. And so, they have a very kind of stereotypical architecture. They tend to have these sort of flanking inverted terminal repeats that mark the boundaries of the mobile genetic element and provide this kind of hairpin structure that tells the transposase enzyme basically where to bind and excise so that it can further mobilize around more genomes. They'll often also have these cargo genes that they kind of hitchhike around the genomic galaxy with and provide some sort of a fitness benefit to the host. Um, there are these insertion sequence elements, which are much more minimal autonomous versions of transposons that, basically don't contain a bunch of these extraneous cargo genes, but just have the transposase enzyme alone that they need to mobilize. These IS elements are incredibly diverse, and there's an entire community that's dedicated to their classification, as you can see on the right, called IS Finder, and they're found throughout bacterial and archaeal life. So they're incredibly widespread. So a few years ago in the lab, we started looking at a particular subset of these IS elements known as IS-110 that were kind of interesting. They had a unique catalytic chemistry for their transposases, these DEDD catalytic residues, and prominently they didn't have these inverted terminal repeats that mark the boundary of these elements. And they contain instead just the transposase that you need to continue to mobilize, and then in orange, these left end and right end non-coding flanks. And so, when we were analyzing these bioinformatically across the IS element superfamily, we noticed that the IS-110 elements had the longest non-coding flanks of the entire kind of IS diversity. And so naturally kind of became curious about their function. And so relatively little has been known about these IS-110 elements in the sort of linear kind of inserted form. We basically have known from old biochemical studies done decades ago that they basically cut themselves out into this circular intermediate and then paste themselves back into double-stranded DNA in order to kind of reconstitute this transposition life cycle. And so one of our grad students, Nick Perry, started to build these synthetic circular intermediates of different IS-110 elements. And what he found was that when you actually circularize the element, what happens is you bring together the -35 box and the -10 box that were previously in the inserted form very far away, and their circular form across the core dinucleotide, which is sort of the element boundary. You basically bring together a canonical bacterial transcriptional promoter, so jacking up transcription. And what you'd expect is turning on this promoter upon reconstitution of this boundary would likely turn on the CDS itself. And so you might make more of the transposon that you need to further mobilize and jump around. But when we did small RNA sequencing, what we saw is that in these golden non-coding flanks, we saw these sequence read pileups corresponding to a cryptic previously hidden non-coding RNA found across 6 different kind of orthologs of these different IS-110 elements. So we thought, what's going on with these? And we decided to take a more detailed look at these RNAs, and we found that they had this very consistent predicted RNA secondary structure with these long exposed unbase pair loops. Now, if you've stared at lots of RNA structures, this would look kind of funny to you because RNAs like to be kind of heavily protected in these hairpins to try to protect themselves from RNases and degradation and attack. And instead, they had these kind of big exposed loops that we found quite strange. And so we wanted to kind of zoom in into figuring out what these might be doing. So we undertook a computational analysis where we could look across thousands of different orthologs of these IS-110 systems and then pull out the kind of subsequences that would sort of consist of this non-coding RNA and target DNA. And we could do here on the top right what's known as a covariation analysis that's more commonly used to look at protein residue covariation and protein evolution, but instead we adapted those algorithms to look at nucleotide covariation. And basically what you can look for is if this nucleotide is changing in the non-coding RNA, is a corresponding nucleotide changing in the target DNA of that transposon. And the actual data was actually extremely striking. When we took the non-coding RNA and laid it out here on this x-axis, and you can see the target DNA on the y-axis, we could see two sort of regions of contiguous covariation that corresponded, when we colored them by expected Watson-Crick base pairing, corresponded to the bottom and the top strands of the target DNA. And so this was the first smoking gun that this might be some sort of RNA guided or potentially programmable transposon. And that started to get us pretty excited. The second thing that we saw was much more surprising, which was on the 3' end of the same guide RNA or non-coding RNA, we see the same contiguous covariation and Watson-Crick complementarity with the donor DNA, sort of implying that this could be a potentially bispecific guide RNA that recognized actually two distinct molecules consisting of the DNA target site, but then also the circular intermediate, which is, of course, the IS-110 element itself. So mechanistically, this seems to be a novel form of RNA-guided self-recognition by a mobile genetic element. This got us really excited about what this could mean potentially for bioengineering. When we took those regions of covariation and projected them onto the RNA structure, we found that they were smack dab in the middle of these exposed unbase paired loops. We named this bispecific non-coding RNA the bridge RNA, inspired by its natural and bridging together target and donor DNA molecules. And so we decided to test this experimentally. We found that when we recombinantly express IS-621 protein, which is a particular ortholog of the IS-110 system, and you in vitro transcribe this bridge RNA, it binds at low nanomolar affinity. And this recognition is also sequence specific. When you add in target DNA or donor DNA, it also binds at low nanomolar affinity, but in a sequence specific way. So if you scramble the TBL, the target binding loop, or the DBL, the donor binding loop, you basically abolish recognition, which you can see on the bottom right. And experimentally, this was sufficient for in vitro recombination in a test tube, which we could verify using kind of traditional biochemical means. So the reason why this is interesting to us is because it implies the ability of the system to do recombination without significant crosstalk with cellular DNA repair or host factors, since we could kind of get it all to work in a test tube, implying that it does the single step insertion reaction and kind of made us excited about the potential to reconstitute these inside of cells. And so this allowed us to basically build a base pairing model where this bridge RNA is able to bind to staggered stretches of target DNA, shown in blue, and donor DNA, shown in orange. We wanted to go ahead and try to verify if this were actually the case. And so we collaborated with a longtime structural biology collaborator Hiroshi Nishimasu at the University of Tokyo. And basically what you can see here is the target DNA and the donor DNA and the active synaptic complex of the IS-621 system. And we've abstracted away the bridge RNA and any of the IS-621 proteins simply to show you the DNA. And what you can see is that the top strands are actually nicked by the system strand exchanged to create an intermediate holiday junction that's then resolved by bottom strand nicking in order to give you the final kind of ligated complex. When you bring in all of the different proteins, you can actually see this very complicated but kind of beautiful mechanism where there's sort of two monomers that bind as a homodimer to the to the tops to the target DNA, the target binding loop of the bridge RNA, and then on the bottom you have another set of another homodimer that binds to the donor DNA and the donor binding loop, creating this kind of homotetramer that gives you sort of four hands that can kind of exchange each of the strands of the DNA. We wanted to take this kind of system beyond in vitro experiments, so we decided to reconstitute them in E. coli, and we basically developed this plasmid or episomal recombination assay where P donor can be recombined into P target in a way to basically give you a GFP gain of function. And what we found was that basically when you can get in this flow plot GFP activation with the wild type recombinase, but when you basically catalytically inactivate the catalytic mutants of the recombinase protein, or you basically abolish the RELE, which is going to give you the bridge RNA from this reconstituted promoter, you can basically isolate in cells all of the kind of key components. You need the active recombinase and you need the active bridge RNA. We then showed that you can reprogram every single base in this target binding loop to actually target the transposon to different nucleotides and insert into different sort of recognition sequences. And that this was able to insert genome-wide in the bacterial genome with insertion specificity of kind of over 94%. The donor binding loop was the next thing that we tried to mutagenize, and we showed here you could also change every base in the donor binding loop in order to reprogram the target and the donor recognition separably and modularly, which got us pretty excited about potential engineering applications. So Cre recombinase is obviously a kind of classic molecular biology enzyme that's been a foundational toolkit of mouse transgenesis for decades. And we commonly have these conditional reporter animals where you can kind of lock stop locks cassette in order to kind of turn on a Cre driver in certain types of tissues. And it turns out by controlling the directionality of these 32 nucleotide invariant LOXP sequences relative to each other, you can actually control the directionality of recombination. So you can get, insertion, obviously the excision that Cre is famously used for, but also by inversion, simply by changing how you flox a locus, right? And what we showed here was that the ability to separably reprogram the target and donor recognition of this bridge RNA would allow us to not just do programmable insertion, but also excision and inversion inspired by these natural transposon life cycles of Cre, simply by giving the right bridge RNA for the desired rearrangement kind of gives you this universal mechanism and unified way with this one molecular system for the types of DNA rearrangements that you want to do. So we reported this work last year in vitro and in bacterial cells, and as you can imagine, a common question folks ask was, does this work in human cells? And so we spent a lot of work trying to make that possible. And so, in the context of mammalian genome engineering, you can think of this as a two-component system, right? You have a bridge recombinase that you deliver as a single chain enzyme, although it can kind of homodimerize to form, active synaptic complex, and then a single bridge RNA that recognizes the target and the donor molecules. And by kind of changing where the target and donors are being bound to relative to the bridge RNA, you can basically control whether you want to do insertion, excision, inversion, or translocation. I'll simply note that in the insertion setting, you need to deliver a double-stranded DNA donor in order to insert into a target chromosome. And this actually leaves a sort of a two nucleotide scar, which is the kind of duplication of the core dinucleotides, which we can talk more about. But for the other three types of rearrangements, it's completely scarless. This is sort of kind of in contrast to other technologies that can lead to long target site duplication and genomic scars of kind of 20 to 50 nucleotides. You can kind of imagine in a therapeutic context this can be much more minimal, but also the RNA-guided excision and inversion don't require a DNA donor. And so in a therapeutic setting, you can do this with all RNA delivery simply by delivering an mRNA that encodes for the recombinase and then a bridge RNA as well, right? And so you could do kind of all RNA LNP type of settings without kind of DNA delivery with an AV donor or some sort of chemically synthesized version of DNA from an Aldebaran or what have you. Okay, so how do we actually get this to work in mammalian cells? Well, we did the pretty obvious experiments where we took the I621 system that I just showed you and put them into human cells. And well, unfortunately, that didn't work. And so, we're like, okay, well, now what? Well, let's go back to the computational drawing board. And we developed a systematic metagenomic analysis of the bridge RNA phylogeny across bacterial and archaeal life, and then essentially did systematic ortholog screening in the human cell context to try to find systems that might work. We additionally did two-component engineering of this system of both the recombinase with deep scanning mutagenesis and then structure-guided bridge RNA engineering. And you can kind of see a high-level summary of what we did here. This is pre-printed as of a few weeks ago. And this was, I guess, a very high-level summary slide, but this was, I will say, a tremendous amount of work to get us a system that can insert kind of up to 20% directly into the human genome with 10x efficiency relative increase in efficiency relative to bridge editing out of the box with the kind of bridge and recombinase engineering. You can kind of get a significant improvement, which you can see with the green star. Beyond insertion into the genome, we also wanted to test excision or inversion. And the efficiency of this is pretty variable, but strikingly, we don't see like a clear sort of degradation of efficiency relative to the length of the manipulation. And in fact, we were able to get nearly kind of a 1 megabase inversions of the genome at kind of reasonable single-digit efficiencies, which of course you could in principle select for with a selectable marker or so on. So we're very interested in trying to understand this and the determinants of these efficiencies relative to things like high-C data and kind of other ways of trying to make this a slightly more predictable method. And so in addition to trying to figure out the length dependence of these changes, which we can do at nearly the megabase scale, we also asked in a kind of therapeutic proof of concept type setting if we might be able to do a repeat collapse, for example, in Friedreich's ataxia, which is one of many repeat expansion disorders where expansion of these nucleotide repeats can lead to different types of neurodegenerative or neuromuscular types of disorders. And basically what we see here is that bridge RNA-based excision is basically able to do repeat collapse of these expanded repeats, which we're kind of now advancing into kind of more primary cell and in vivo settings. So in summary, we kind of think of this kind of bridge recombination system as a third generation of native RNA-guided systems. They retain the key property of programmability of RNA interference and CRISPR while kind of enabling functions beyond merely RNA and DNA cutting. And we think this is an important step for biological programming because you can universally insert, cut out, or flip any two pieces of DNA, from in vitro all the way to the human genome directly. So with the ability to make these large types of changes, one of the questions that you start to ask yourself is, what do I do with all this size, right? And gene therapy, we tend to have this, at least especially for monogenic disorders, this fairly straightforward paradigm of I'm missing a gene and I need to put it back, right? But if I had the ability to put in, much more DNA or make much larger types of rearrangements, what is it that I would want to do? And so this has sort of been recognized by Frances Arnold, the inventor of directed evolution, where she sort of said in a talk, today, for all practical purposes, we can read, write, and edit any sequence of DNA, but we cannot compose it. Maybe we can cut and paste pieces from nature's compositions, but we don't know how to write the bars for a single enzymic passage. And then she ends by saying, however, evolution does. And so we started thinking, well, how can we learn the language of evolution? So machine learning is starting to give us this potentially universal paradigm where we can learn complex higher order patterns from tremendous amounts of unstructured data, which of course works in natural language, which with things like ChatGPT, but increasingly we're seeing advances of generative AI in vision or even in embodied intelligence like in robotics. We thought, could we take these types of kind of modern transformer-based approaches and apply them to biological data? Then machine learning is also starting to revolutionize biology, where models such as AlphaFold or ESM enable protein structure prediction or design, the prediction of protein-protein interactions or protein-ligand interactions, or even to predict the complex folds of RNA. But despite these advances, the complexity of these molecules is really dwarfed by the overall complexity of an entire cell, which is many orders of magnitude larger and more complex. And so the cell's structure and its function are built up by the diverse interactions between all these different molecules, which is an incredible number of combinatorial possibilities at multiple levels of regulation. So as all biologists know, in short, this is really, really complicated. The good news is that all of this molecular logic is encoded within DNA, which is a single sequence that contains all of the information that's needed to arguably create an entire organism. And in fact, the effects of natural selection are passed down throughout generations of life via these DNA mutations. And so the question that we asked is, could you learn something about the function and the functional adaptations to natural selection by looking at DNA alone? And so in sort of this kind of Turing machine kind of concept, we asked this simple question: can we create a machine in this sort of computer science sense that learns information from these whole genomes to try to model the kind of complex interactions of these molecules that can create an entire organism? And so one of the central challenges in doing this is that biology, of course, uses a different language. It's foreign and not very human interpretable. And so you can see this kind of famous schematic of the central dogma of molecular biology, where DNA is transcribed to RNA and translated into proteins. But you can see is that the sequences are continuous and directional, and that there's actually information flow that's ordered as molecules predictably flow across these different modalities of the central dogma. And so, there's actually a tremendous amount of structure that's embedded inside of these, otherwise kind of, confusing and inscrutable sequences. And so to try to decode this molecular language, right, you can try to, use the transformer paradigm from kind of modern deep learning to try to decode or translate any type of language. And so if you take this famous saying from human literature, right, to be or not to be, right, we all know the next word or the next token, right, where we can do next token prediction. But if I ask you GTG, CATCT, right, would you know what the next nucleotide is, right? And to give you a sense of why next word or next token prediction is incredibly general, it's useful to look at a few examples. If I said in my free time, I like to code and not I like to banana, right? You might learn a sense of grammar. Okay, that's very straightforward. That was why these tools were invented in the first place, to try to do translation and natural language processing type tasks, right? But you can also learn things like world knowledge. The capital of Azerbaijan is Baku and not London, or I'm giving this talk in LA and not in Portofino, right? You can learn a sense of where you are. But even more deeply, you can get a sense of things like spatial reasoning. Iro went into the kitchen to make tea. Standing next to Iro, I pondered my destiny. Right? I probably then left the kitchen and not the bathroom, right? And so with massive amounts of text, you can learn a sense of vision and navigation, right? And these concepts extend to biology, where for a long time we have tried to understand how genotype and phenotype is related, where a genotype can try to underpin the mechanism of phenotype, which is in turn selected functionally in order to influence the genotype, right? And so this sort of intuition of complex biological properties can actually be deconvoluted by looking at sequence variation alone, right? So if you basically look at a stretch of nucleotides, you can see if the next base in my genome is going to be healthy or is going to give me a deadly genetic disease. At the level of residues, the next residue might be the difference between a catalytically active binding pocket or a null molecular machine that doesn't do its important function in a cell cycle or tumor suppression, or the next nucleotide in an RNA structure might give me the right hairpin that's the functional aptamer that can be used to recruit transcription, or it blows up the hairpin and then I basically don't get any biological activity. But even at the abstraction level of genes, right, we know in prokaryotic genomes, functional complexes are often coded in cis sequence into functional operons. And so if I have mod A and mod B in an operon, the next gene is probably mod C and not something like rec A, right? And so we trained such a model called EVO1, where previously in the field, folks have trained biological sequence models that are specialized for a particular modality of the central dogma. So you might train a protein language model by looking at billions of protein sequences, or a codon language model or an RNA language model by looking at those sequences in particular. Our key insight was that all of these different types of modalities are embedded fundamentally within the genome alone, and that we might be able to learn about them by training long context genome foundation models that can look at all of these elements with attention, right, at the same time. And so the first thing that we saw was a very interesting observation that we've seen in other fields of deep learning, like vision, where basically larger models have better performance at this task of next token prediction. And we showed that basically here too for DNA for the first time, implying that similar strategies of scaling, where larger models, more pre-training data, more compute poured into the model, would all be fruitful paths to improving genomic language model performance. And so this first model that we trained EVA1, we basically showed that it's both predictive and a generative model. We can do things like generate functional CRISPR systems, which require a Cas enzyme and a non-coding RNA. Protein language models, or things like AlphaFold, don't natively understand how a non-coding RNA or protein might talk to each other. It only understands the activity of proteins. And this EVO, by looking at DNA alone, understands something about the coevolutionary relationship between these two different types of molecules in order to compose systems that can actually bind to each other and actually be experimentally functional. And we actually showed in our study that this could work as well as the state-of-the-art Streptococcus pyogenes Cas9 and cleavage assays. At the genome scale, you can do things like simulate the effect of CRISPR screens in silico, right? Where, for example, many folks do gene essentiality CRISPR screens that can, be very laborious and take students, months to a year in the lab. Here, we can basically kind of classify genes based on their essentiality with forward passes of a neural network. And I think in the future, it will be exciting to see how these types of models, as they get better, might start to replace certain things that would be traditionally the domain of the wet lab. Even kind of more kind of speculatively, we show that the model could generate sequences potentially even at the genome scale. We can make sequences, a megabase in length that contains proteins embedded within the genomes that we generate, that you can see have a dense coding architecture on both strands, contain proteins embedded within those genomes that map to fundamental ontologies like ATP binding or DNA binding transcription, and actually have all of the kind of rRNAs that you need, but unfortunately, not all of the it has all of the tRNAs that you need to do protein translation, but actually, interestingly, not all of the rRNAs that you need to translate those proteins in turn. And so it's sort of, we kind of think of this like a blurry picture of life. It's sort of an interesting kind of step towards a future where you can do whole genome design. But of course, if you made these today, they wouldn't, be viable, we imagine. But we're kind of excited about this as a multimodal and multi-scale model that unifies individual molecules, can design molecular systems, or potentially even make things at this scale of whole genomes. So this model EVO1 was trained on prokaryotic genomes, right? So on bacterial and phage genomes that are short, and allows you to make much more computationally efficient models. Um, and for EVO2, of course, we wanted to try to look at multicellularity, including human genomes, which of course are much larger. This is much more computationally intensive, but then also has these long non-coding stretches that can be governed, under very different types of kind of selective rules. And so to do this, we trained EVO2 across all of observable evolution, so across kind of all three domains of life: bacteria, archaea, and eukarya, on over 9.3 trillion tokens of DNA. So at a scale of pre-training data that's comparable to kind of current generation language models. And we extended the context of this model to 1 million base pairs, in contrast to the 131,000 base pairs from EVO1. The reason why we cared about doing this was because, of course, you can look at individual molecules with pretty short context, but at 1 million bases, you can move all the way up to the scale of entire eukaryotic chromosomes, entire minimal bacterial genomes, or things like human TADs, topological associated domains. And by looking at all these different things in context, can you learn about the kind of higher order structure of the genome? And we scale this to 40 billion parameters. So we, to do this, had to invent a new type of machine learning architecture known as striped hyena 2 that basically stripes, as the name suggests, layers of kind of dense attention from transformers like in ChatGPT with these so-called hyena blocks that use gated convolutions that are able to evaluate very long context information in a more computationally efficient way. And the reason for doing this is because we don't have the compute budget of a research lab like DeepMind or OpenAI, and so you kind of have to pioneer new model architectures that can be computationally efficient to be able to kind of train on a multi-trillion token corpus of, in this case, DNA. Okay, that's cool. What does the model actually do? And one popular area of computational biology is to do a variant effect prediction, where if you have mutations in your genome, you might want to understand, for example, in BRCA1, whether or not my mutation is going to be pathogenic. And for example, in causal, some folks have causal variants, they might elect for a double mastectomy. Or if you have a benign variant, you might elect for an annual mammogram. But most mutations are these kind of variants of unknown significance, which is kind of fancy science speak for we don't really know what's going on, right? And so it turns out when you put these mutations into the model, Evo has an opinion on whether or not that model will be pathogenic or not. And it does so in a zero-shot setting that is state-of-the-art for non-coding mutations, and just below alpha missense from DeepMind for coding mutations. And I'll simply note that alpha missense was explicitly trained on all human genomic variation, whereas EVO2, interestingly, has only seen one human reference genome in training. And in fact, it's learning its opinions about mutational pathogenicity by learning about the expansive mutations across fish and fly and monkey and whatever genomes, which we think is extremely interesting. And we have shown these evaluations zero shot, where the model is simply given this example and asked to evaluate it, but the kind of latent space of these models of course, contain much more information. And so you can actually train a supervised model on the embeddings from evo2 and actually recover state-of-the-art activity. We're, of course, injecting the model explicitly with human genomic variation to try to understand how we can kind of make this a generalist kind of state-of-the-art model for variant effect prediction. So one of the common criticisms of these models is, okay, they're these black box neural networks, so the models are learning something, but what are we learning? And so there's this field of machine learning known as mechanistic interpretability. They use mechanism in a way that's inspired by the way biologists talk about mechanism, but the goal is to try to take these large black box language models and break them down into observable human component, like human interpretable components, right? And so from the Anthropic research lab that trains the Claude series of models, they've sort of done this mechinterp work where they found semantic features that correspond to the concept of the Golden Gate Bridge, which you can see lights up on a bunch of different languages, showing generalizability of this feature. And so interestingly, if you ask Claude, what is your physical form, it will sensibly tell you it doesn't have one. I'm simply an AI and a software avatar. But if you actually manipulate the weights of this Golden Gate semantic feature and clamp it to 10x its weight, it now becomes obsessed with the idea of a Golden Gate Bridge. It'll tell you I'm a Golden Gate Bridge, and if you say, okay, that's great, but what is the Fibonacci sequence, right? It will start to tell you something about math, but then inevitably start to tell you about how it's actually really lately been thinking about sweeping vistas and golden kind of things. And so it kind of brings the conversation back to the thing that it's obsessed with, maybe like talking to an old professor. And you can try to take these ideas and apply them to genomic language models. And so we trained similar sparse autoencoders on evo2 and to try to learn what it might be learning about biology. And I'll simply note, evo2 has never been told anything about biology. It doesn't know what a gene is, doesn't know what a pathogenic mutation is, it doesn't know the difference between a fish or fly or worm or a human. It simply knows how to predict the next base. Okay. And in fact, when you train these sparse autoencoders, you can find features that light up genome-wide across the E. coli genome, represented here in a plasmid, where you can find features that correspond to prophages, which are bacterial viruses that have inserted themselves into genomes, but light up not just for prophage sequences, but also light up on the kind of CRISPR spacers that are acquired in its process of adaptive immunity to kind of fingerprint invading phage. And so somehow it's learned the concept of these sequences being foreign. We can also find features that correspond to open reading frames, intergenic regions, tRNAs, and rRNAs. But intriguingly, it finds higher order features like alpha helices or beta sheets, of course, because proteins are fundamentally embedded within genomes, but it's able to find multimodal features of proteins by only training on DNA alone. We can find beyond prokaryotic genomes things like features that seem to fire on transcription factor motif-like sequences. And this is again in the human genome now, looking at non-coding sequences upstream of transcriptional start sites. You can actually see things that seem to correspond to TF motifs and also to eukaryotic genome architecture like UTRs and coding sequences and introns and exons, but intriguingly, not just an intron or an exon, but also the leading and the lagging edge of an exon, right? And we can use mechinterp not just for finding basic obvious things, but it can also find things like errors in computer code, which is very useful for people like me. But it can also find errors in computer code. And it actually has an opinion on frameshift mutations and PTCs over kind of things like synonymous mutations. And this is actually super consistent. For example, we looked at this feature across 100 different genes in the human genome. So beyond prediction and interpretation of genomic language models, I kind of alluded previously to generation. And so, we showed in this sort of larger scaled up version of Evo that we might be able to try to generate things. And here we showed that we can generate the, human mitochondrial genomes, for example. In the dashed box, you can see your mitochondrial genome. The other 6 lines are Evo generations that aren't simply memorized and regurgitated, but actually can preserve all the key PPIs in the electron transport chain that's embedded within the mitochondrial genome. And we're now making these and actually going to insert them into cells and see what they do. For bacterial genome generation, we actually saw some evidence of potential scaling laws where with EVO1, you kind of get this blurry picture. With EVO2, you get much higher quality proteins embedded inside of these genomes that you generate that sort of conjure the kind of scaling laws that we see with image generation, where you might be trying to make this friendly kangaroo with sunglasses that says welcome friends, but instead, kind of in EVO1, maybe we're seeing this raccoon-like thing and the mission of San Francisco, with this kind of uninterpretable sign. But with kind of more data and more scaling, you can kind of sharpen its sense of its training data. We can generate, entire eukaryotic chromosomes as well. But folks often ask with these DNA language models, well, this is all really cute, but what about epigenetics? And so we showed that a generative model like Evo can be given an oracle like Informer, which is trained on ChIP-seq and DHS data, and it has a sense of chromatin accessibility. And you can basically generate wide peaks or narrow peaks, and by basically pouring in more computer inference time, you can actually make epigenetic sequences that conform better to the user specified pattern based on the amount of compute that you pour into the model. So this sort of kind of more thinking time seems to give it better genomic design. And this allows us to encode fun things like Morse code for ARC or EVO2 in these computationally predicted epigenetic patterns. We've now generated these and knocked them into living cells and done a tax-seq, and you actually reconstitute these patterns very cleanly. But beyond simply epigenomic design, this is actually a very general concept where you can have a generative model hooked up to any scoring function beyond epigenome structure, like generically some sort of sequence to function predictor. So we think of this the sort of series of foundational models as potentially creating an app store for biology across the epigenome and RNA and protein and cellular levels of biology. We showed a few examples of these in our study. So I'll just sort of try to kind of wrap up by saying the next steps from here are to take these large pre-trained models and RL them, right, with active lab in the loop data so that we can take biological feedback and give it to the model so that it can actually improve the quality of its predictions or its generations. And by generating these genome scale or megabase scale sequences, right, we think we can move beyond gene editing or base kind of pair level changes or gene level changes to kind of much larger scale genome design. In the broader sense, biological discovery is basically this search problem, right? Where we're basically trying to do guess and check, right? Where you have an idea and you want to test it and you do an experiment to test it, but this is very kind of low throughput, right? And so what this sort of feels like is if you look at the night sky and you're trying to find the constellations and like where is the Big Dipper and where's the North Star, and I'm not really sure. And actually what I want is an app, like the Night Sky app that just gives me this co-pilot that tells me where I'm looking at in space and what kind of structures I'm seeing. Since biology fundamentally is orchestrated across space and time, to build predictive models of the cell, right? You would want something that can move beyond genomes to also understand universal cellular representations to be able to predict how cells can dynamically respond and transition between states, right? And the goal for doing this is, in principle, you could invert such a model and ask it to say, what are the factors that allow me to click and drag cells from a degenerative state to a hyperexcitable one, or from a hyperexcitable state to a homeostatic one, right? And that's fundamentally what we do with drug perturbations, which are antibodies or small molecules that bind a particular factor and try to turn it off, right? But the combinatorial search space is incredible. And there might be 8 different changes in some temporal order that you need to do to move cells between states. And we have examples of this in reprogramming, in transdifferentiation, knockdown, and so on. And can we have a model that figures out the kind of combinatorics of this? And we're trying to tackle this systematically across both the data side and then the modeling side. And so we've actually developed AI agents that can search the sequence read archive to generate actually what is now the largest atlas of single cell data in the world, where we have nearly 500 million cells that's actually been scraped from all of the 10x data that's ever been generated and deposited to a government sequencing database, something you can never convince a human to do. But an AI agent will just kind of crank on this indefatigably. So, for all the discussion about, AI and work and the future of jobs, right, it seems to be willing to do the stuff that, no one would actually want to do in the first place, which seems pretty good, right? And anyway, like even if you pre-train these data, right, you need to be able to develop kind of data-specific models or active learning with the models to learn biology. And we're trying to tackle this end to end, and we'll be kind of releasing some of our kind of first virtual cell models shortly. So anyway, I hope that I've kind of convinced you that to complement the ability to read the genome, we need to kind of complete the loop of this biological Turing machine, where we have AI models that can help us think and reason about biological state, and technologies to write those programs or sequences back into cells. So this is sort of some of the work that we've been really excited about doing lately. I'll just sort of end by kind of thanking the folks. EVO2 was an enormous team of many researchers, as you can see here, across the ARC Institute, our partners at NVIDIA, various folks at our partner universities, and the MECINTERP lab, Goodfire. And the bridge editing work was led by Matt Durant and Nick Perry in the lab. The megabase scale human genome editing work was done with Silvana Konerman. The bridge editing structures with Hiroshi Nishimasu, and the EVO models with Brian He at ARC and at Stanford. Thank you very much. I'm happy to take questions.
Perfect. And I'm sure there's going to be questions from the audience. That was really quite spectacular for us. And I'd throw it out to the audience for questions at the moment. And if not, I'm going to ask the first question, and then I'll look at things online.
Thank you. Well, I think we're looking at the future. Epigenesis seems to be on your target range. Do you have any comments about offhand what we should be expecting there?
Epigenetics are in our target range. What would we be expecting? Can you ask that a different way, or what do you mean by that? Yeah.
You're putting me on the spot here.
Oh, sorry. Yeah.
It seems to me that gross generalization would be that some of the stages in development that we used to call it determination are epigenetic states of the cells. The transition between those cells that could be pluripotent stem cells from somatic cells, as an example. Mm-hmm. And that transition seems to be within your range of describing what's actually going on.
I think the way that I think about this is the classical way that we try to describe biological mechanism involves compressing very high dimensional data into something very simple that we can put into figure 2A, 2B, and 2C, right? And then you build some sort of model. For example, in genetic epistasis, you might have A, arrow, B, and then some flat thing that inhibits C. But we of course know that these networks are not independent of each other, and there's actually some kind of highly kind of you can represent it as a graph or as some sort of higher dimensional embedding space, but it's a very combinatorial type of interaction. Yeah, I think epigenetic states and the cellular states that the epigenetic signatures are trying to represent are, I think, similarly high dimensional, and we might sort of hope for two, I guess, practical things from these types of models. The first is, could you, without any inductive bias or pre-knowledge, do things like rediscover Yamanaka factors or transcription factors that are capable of specifying cell fates like ASCL1, neurogenin2, MyoD, etc. Can you find those individual factors that feels very within our grasp? And I think these types of biologically informed evals will be critical to convince wet lab biologists that any of these AI models are useful or interesting in the first place. The other is to find something like OSKM, right? Was sort of kind of originally done, whether by your Yamanakas or your Jamie Thompsons or whatever, in some sort of like combinatorial screening, right? Sort of manually done by a bunch of postdocs. And if you want to find, if you have, let's say, 1,600 transcription factors and you want to screen the kind of triplet or quadruplet space, right? That's billions of combinations. And can you build something that's an experimental co-pilot for narrowly sort of giving you the top 24 things to try? So I don't think we're in the era today where these models are sufficiently good that they'll do anything that zero shot out of the box, quote unquote. But I think informed with kind of information and knowledge about what you're trying to do, I think they'll be very useful. I think right now we're training these models. Like Evo is a model that's DNA in, DNA out, right? And so it's not the most approachable, intuitive type of model. And one of the things that we've been exploring in the lab is how to basically make Evo type models accessible with natural language models. So can you use a ChatGPT style interface to basically just tell Evo what to do? And it has information from the literature by pre-training on all of PubMed, combined with information from training on nucleotide next token prediction to improve its performance on variant effect prediction. There have been recent work from our art colleague Hani Gadarzi showing this is actually the case. And by showing it explicit examples of ClinVar, right, you can actually improve its ability to do VEP prediction, without, by simply kind of training on ClinVar data alone. So I think there's just like lots of angles for exploring how we can make these things actually useful to folks so that you can access its knowledge or tool call it as a practicing researcher yourself, as opposed to simply kind of reading individual academic studies. I think the kind of future of this is, right now, if you want to do any dry lab workflow, right now you kind of are doing tool calling yourself, where you read some papers, you think about it, you do a blast, you do an AlphaFold prediction, you think about it, you talk to your PI. But like, what if this could all just be done end to end, where you have a reasoning model, you ask it to look at this thing, you give it the count tables from your flow cytometer or the FASTQ files from your MySeq, and then it you give it the DMUX labels, and it just looks at all the data and finds the patterns for you with this automated data scientist. I think that would give every one of us a lot more leverage and allow us to move much faster in research. And I think folks would be extremely surprised at how close this is.
Thanks, Patrick. So could we ask you just in one domain, just in terms of the practical issues of what you're doing? So across the issue of, lots of people are getting their whole genome sequence. We do that for Mendelian genetic diseases.
Yeah.
And we often can't solve it, right? So, and where is it that we should look?
Yeah.
So just taking your EVO2 model, what's your projection of what that sort of additional positive predictive value of being able to possibly interpret the non-coding RNA, for instance? Do you think a lot of the answers are there, and now it's much clearer where to look in the genome? Or give us a practical sort of flow.
Yeah.
How would we apply this now?
Yeah, the evals that we did here were for individual VEP, where we use ClinVar as the eval data set, where we have individual mutations in, you know, all of the different genes.
But those
and
are
the
protein
label.
coding genes
Right,
that kind of
right.
have the structure of reading
Right.
frame, which makes them easier. So when you're in
Right.
the more amorphous non-coding RNA,
Yeah.
which is exciting, the rate
Yeah.
that you're sort of.
I think it does. It's state of the art in non-coding VEP. I don't think that means it's really, really good, right? And obviously, this is also only for individual mutations and not for combinations of mutations. And I think that's the important step that we need to do in ongoing and future work, is to basically say, at what point is this as good as PRS scoring, or it can actually even be better? Like, for example, you know, these human genetics models explain maybe only 30% of the genetic of the variants in like profound autism or something like that. And you know, this model, of course, is trained only at 1 million token context. While that's really good for, I think, biological language models, certainly today, right? The, you know, mutations might be more than 1 million bases away, right? And so I think the future is maybe you have 3 billion context models. Like the latest Llama 4 model from Facebook is now at 100 million tokens. So that's, you know, 100x better than what we did here, obviously, with, you know, much more than 100x more compute and people than we have. But you know, we'd like to get to the frontier of what folks do in Gen AI in these biomodels. Yeah.
Very good.
Hi, thank you for the talk. One question that I had is, this next token prediction is obviously very, very good. And as you said, as the token length increases, that will get better. But one concern is also identifying those entities. So these entities, like, is it a gene, or like explicitly knowing this is BRCA and being able to have that causal reasoning?
Yeah.
A lot of people have been trying to turn to, for example, neurosymbolic models.
Sure.
Do you see that as an example that you guys could head to, or something similar or different?
Yeah, I mean, I think like, you know, there's obviously this extensive academic tradition of causal ML or interpretable ML, or you know, alternative architectures that might be more white box. I think those are all, I think, very useful and should be used depending on the biological problem that you're trying to solve. There's a lot of tailwind to try to make these types of transformer-based models much more open box with advances in things like mechinterp, which is why we invested a lot of time and effort into that here. But yes, for sure, you can give models like Evo a lot more inductive bias and, you know, fine tune it against an explicitly labeled set of examples that, you know, kind of teach it explicitly about promoters, or explicitly about protein coding genes, or exon intron junctions, or splice mutations, or what have you. And so, you know, I think there are lots of ways that folks are going to be and are already using these models for specific experimental use cases. Yeah.
This is amazing work. Thank you. My question is that like some help to understand what the 10x single cell data trained model can do. So let's say if I make an example, maybe you can tell me if this makes sense. So I work on neuroscience research. Let's say the goal is to discover new drugs for Alzheimer's disease. So your model is already trained on a lot of 10x single cell data, and now I give the model a Alzheimer's single cell data and the control single cell data, and I want the model to predict what are the key genes that could help. And then we would validate these key genes by knocking them out in mouse models and then in human clinical trials. Do you think this is like a future that you envision how this model will
Well,
work?
that's like a pipeline of many different steps, right? No AI model will do in vivo experiments and IND enabling studies and GLP and tox and what have you, right? But I think explicitly what virtual cell models will do, which I didn't describe extensively here today, initially a lot of work is being done on things like perturbation prediction, right? And so yes, let's say you have these, virtual cell models pre-trained on single cell atlas data, right? And then you have a bunch of AD human genetic studies where you've linked the variants to different genes, right? The questions might be, do these mutations affect these genes in astrocytes or in microglia or in neurons? And what is the actual cell type that these variants are acting through? I think the model could be very useful in understanding that. You might also be interested in understanding the epistatic hierarchy of different pathways in the single cell context. So, if there's some sort of, two sets of inflammatory pathways that are, kind of, I don't know, TNF and NFKB or whatever, and sort of if I activate TNF signaling, what happens to, this part of the NFKB? Is it up or down, right? These types of models will hopefully help us do that type of stuff. Yeah. So that's just a more granular sense of how you can actually use these models in the future.
Thank you.
And you have one question from online, which we'll get to. It says, and I'm going to guess they're thinking of viruses here. Can you speak to the ethics of genome design and how safeguards against accidental gain of function are being encoded in the model training?
Yeah, no, it's a wonderful question, and we took that very seriously in this work. And explicitly, we've withheld eukaryotic viruses from model training and have a bunch of supplementary figures evaluating the model and viral generation. So to show that it's very hard to you cannot make coherent SARS-CoV-2, for example, genomes from EVO2 because it's just not seen this in training, so it doesn't kind of understand it, and it would have to do so like very out of distribution. Of course, this is an area that's very hot in Gen AI, where you might ask ChatGPT or Claude, how do I make anthrax, right? Or how do I I don't know. You have all your own ideas, I'm sure. And the model is aligned, to not do this, and it's been kind of given a bunch of human labeled examples in the RL of the model to kind of not advise you granularly on how to enact criminal activity. But of course, there's lots of work to jailbreak these models and kind of extract that type of information, which is absolutely in the latent space of these models because it's trained on, I don't know, 4chan or something. And so, can you jailbreak biological language models? I think that will be a cat and mouse game for years to come.
All right, very good. I think we've run over because of all the interest in your thing. So I think we should all give Patrick a
Thank you very much.